Welcome back to this lesson on translation. The learning goals of this session are 1. Understand why transcription and translation are coupled in prokaryotes. 2. Describe the 3 stages of translation with illustrations. 3. Identify the 5' and 3' UTRs in mRNA. And 4. Define polysome and state its advantage. I would begin by reminding you that translation is the second step of gene expression. It refers to the process by which a protein is synthesized from an mRNA. As the name of the process implies, translation involves translating information in one form of language into another language. To put it precisely, I mean that the language of nucleotides in mRNA is translated or expressed into the language of amino acids. The instructions for a polypeptide or a chain of amino acids are coded in the sequence of A's, G's, C's and U's of an mRNA. Now, to decode the mRNA and synthesize a polypeptide, 2 other key players interact with mRNA. And they are tRNA, which brings correct amino acids according to the sequence of mRNA, and ribosomes, that are the structures where protein synthesis actually take place. Above all, cells that make more proteins have usually higher number of ribosomes. Remember that the cells in our body are constantly making proteins throughout the day. While you're trying to learn the process of translation at this moment, your cells are efficiently making proteins for you. In fact, by the time you finish watching this video, your cells will have synthesized several proteins. We might hardly realize it though, but our cells and its molecular machineries are taking care of us by doing the job all the time. And as a matter of fact, proteins play a central role in essentially all biological processes. They function as catalysts or enzymes. They regulate gene expression. They help in the transportation of essential molecules such as oxygen. They transmit nerve messages from cell to cell. They also provide structure to cells and organisms. And as you know, proteins such as antibodies protect us against infections and so forth. So do you see what I mean? Well, my point is that proteins are basically found in everything, from cells to tissues to organs, and they are being synthesized continuously by the cells through the process of translation. Despite being a universal process, translation in prokaryotes and eukaryotes differ in certain aspects. One critical point that I want you to know is that translation is coupled with transcription in prokaryotes. This is to say that translation can begin simultaneously from the 5' end in prokaryotes while the mRNA is still being transcribed. And the 2 main reasons why prokaryotes have coupled transcription and translation are: firstly, prokaryotes lack a separate nucleus, and consequently both transcription and translation occur in the cytoplasm. Secondly, the newly synthesized mRNA in prokaryotes is an uninterrupted mature mRNA that can be translated right away. On the contrary, transcription and translation are separated in time and space in eukaryotes. Perhaps you can think of the reason why it is so. Eukaryotes, as we know, possess a distinct nuclear membrane, and so transcription occurs inside the nucleus. The resultant primary transcript in eukaryotes, however, is an interrupted pre-mRNA. As a consequence, pre-mRNA is subsequently processed into a mature mRNA by removing the introns. After RNA processing, a mature mRNA is then exported out of the nucleus into the cytoplasm for translation. Well, the fundamental process of translation in all organisms basically proceeds in 3 stages, which are initiation, elongation, and termination. During initiation, the ribosomal subunits assembles around the mRNA, and the initiator tRNA with methionine amino acid is attached at the start codon, which is AUG of the mRNA. In the second step, which is elongation, the amino acid chain gets longer as new amino acids are being added into the ribosome. And finally, during the termination stage, translation gets ended at the stop codon and the newly synthesized polypeptide chain is released. So with this overview, let me now explain the three stages in a bit more detail. In the first stage of initiation, the initiator tRNA, which is a tRNA containing methionine at its 3' end, binds to the small ribosomal subunit. So this is the small ribosomal subunit with the help of initiation factors. So such a complex made up of the initiator tRNA and the small ribosomal subunit then binds to the mRNA. So let me bring the mRNA. So this is our mRNA strand. So the complex has now bound to the mRNA and the initiator tRNA's anticodon binds or base pairs with the codon AUG of the mRNA. And this is followed by the joining of the large ribosomal subunit, which is this. The large ribosomal subunit then positions the initiator tRNA at its P site and thus completes the initiation complex. So now this whole unit is referred to as an initiation complex. And an important point that you want to remember is that the first tRNA or the initiator tRNA always occupies the P site of the larger ribosomal subunit. And at the end of initiation, the next mRNA codon, now in this sequence it is UUU, is exposed. Before moving to the next step, I want you to understand the sequences in the mRNA which are referred to as UTR sequences. UTR stands for Untranslated Regions. So we have UTR sequences at the 5' end of the mRNA as well as we have UTR sequences over here at the 3' end of an mRNA. So what do we mean by, what are these UTR sequences at the 5' and 3' end? 5' UTR refers to the 5' sequences in the mRNA before the start codon. We know that translation begins or starts from the start codon. So the sequences before the start codon are not translated. Hence they are called as 5' untranslated region or simply 5' UTR. Similarly, the sequences which are referred to as 3' UTR. 3' UTR sequences refers to the 3' sequences in the mRNA after a stop codon. And we know that translation terminates or ends at the stop codon. So the sequences beyond the stop codon which are not translated are referred to as 3' UTR. The second step of translation is elongation. Of all the three stages of translation, elongation in prokaryotes and eukaryotes is the most similar step. Elongation begins with the delivery of a right amino acid into the empty A site of the ribosome by a second charged tRNA according to the mRNA codon sequence. So in this sequence, AUG codon is followed by the next sequence which is UUU. And we know that the codon UUU codes for phenylalanine. So the second charged tRNA to be entering into the ribosome into the A site is going to be a tRNA carrying phenylalanine. After binding to the A site, the complementary anticodon of the tRNA base pairs with the codon of the mRNA. So here the complementary anticodon is AAA since the codon is UUU. Another point that you may want to note here is, unlike the initiator or the first tRNA, the second charged tRNA and the subsequent charged tRNAs are always to be entering into the A site of the ribosome. So it's only the initiator tRNA that occupies the P site of the ribosome. All the rest of the subsequent tRNAs are to be always occupying A site of the ribosome. It is then followed by the most important event of elongation, that is the formation of the peptide bond between the two amino acids. And this reaction is catalyzed by the enzyme peptidyl transferase in the larger ribosomal subunit. During peptide bonding, the enzyme peptidyl transferase will transfer the amino acid from the tRNA in the P site to the amino group of the amino acid in the A site. So in this case, methionine will break off from its tRNA and then form a peptide bond with phenylalanine. After peptide bonding, the uncharged tRNA in the P site is translocated into the E site for exit, and the tRNA in the A site with the amino acid chain is then shifted into the P site. And such translocations of the tRNA is possible when the ribosome moves along the mRNA by one codon. With its movement, the ribosome then reads and exposes a next codon of the mRNA in the A site, and the process is repeated. Let me go through the process one more time. So elongation continues with the delivery of the next amino acid corresponding to the codon of the mRNA into the empty A site. And since the third codon reads as ACC, it is specifying for a tRNA carrying threonine, because ACC is the codon for threonine. So the third charged tRNA to be entering into the A site of the ribosome is going to be a tRNA carrying threonine. After entering in, the complementary anticodon of the tRNA base pairs with the codon ACC. It is then followed by the formation of peptide bond. And as we know, the enzyme peptidyl transferase will transfer the amino acid from the tRNA in the P site to the amino group of the amino acid in the A site. So here, phenylalanine will break off from its tRNA and form a new peptide bond with threonine. After peptide bonding, the uncharged tRNA in the P site will be translocated into the E site for exit, and the tRNA in the A site with its peptide chain is going to be shifted to the P site. And this happens, as you know, when the ribosome moves along the mRNA by one codon. As the ribosome moves along the mRNA by one codon, it exposes the next codon of the mRNA in the A site for a next amino acid. In this manner, the ribosome continue moving along the mRNA one codon at a time in a 5' to 3' direction, and the polypeptide chain keeps extending as specified by the sequence of mRNA codons. And all these elongation events are facilitated by several of elongation factors. In the end, translation is terminated when the ribosome finally reads any of the 3 stop codons at the A site. And in this sequence, the stop codon is UAA. As we know, the 3 stop codons do not code for any amino acid, but signals the end of translation. So a protein called releasing factor binds to the stop codon, and the binding of the releasing factor to the stop codon triggers the separation of the polypeptide chain from its last tRNA. So serine in this case is going to be breaking off from its tRNA, and the polypeptide chain is now separated from the tRNA. This is also followed by the dissociation of the ribosomal subunits, the tRNAs, as well as the release of mRNA. The newly synthesized linear polypeptide chain can eventually fold into a specific protein structure, and the released mRNA, tRNAs, and the ribosomal subunits can be recycled to take part in another round of translation. Well, that's how you can understand the 3 steps or stages of translation. Now, before I wind up this lesson, I also want you to be aware of polysomes in prokaryotes and eukaryotes. Usually, multiple ribosomes can translate a single mRNA at one time to produce many copies of the polypeptide simultaneously. And such a group of ribosomes translating a single mRNA simultaneously is called a polysome. Through the formation of such polysomes, the efficiency of protein synthesis is greatly increased. We can say that polysome saves both time and energy. And this is the reason why we can synthesize protein in just few minutes. That's it on translation, and I'll catch up with my next lesson.